Okay, well, welcome everyone. Thank you for joining today's ASBMR Member Spotlight, brought to you by the ASBMR Diversity, Equity, and Inclusion Committee. The series aims to highlight the research of ASBMR members in a collaborative setting and will be taking place bi-weekly on Thursdays Eastern Time through the end of 2020. ASBMR members are comprised of a diverse collection of individuals from different backgrounds, countries, and cultures who bring a host of life experiences, values, and belief. And this series aims in particular to shine a light on the research being done by ASBMR members belonging to groups underrepresented in biomedical research. All members, however, are invited to participate in these sessions. Highlighting our members' diversity is not only reflective of the world, but will make our community more innovative and produce better scientific research. Today's spotlight will be featuring Dr. Ahmad Othman, Ph.D. candidate at Rush University Medical Center, who will present research examining the role of RUN-X2 in promoting microtubule stability in autophagy and bone metastatic breast cancer. I hope I pronounced that correctly. And just as a reminder, the presentation will last approximately 20 minutes, and following the presentation, there will be an opportunity for question and answer and discussion with the audience. And as a reminder, please just remember to keep yourself muted during the presentation and unmute or use the chat feature to ask questions following the presentation. And with that, Ahmed, I would like to invite you to begin your presentation.
Good afternoon. I'd like to thank the ASBMR for this opportunity to share some of our exciting research with you today. My name is Ahmad Othman. I'm currently a PhD candidate in the Department of Cell and Molecular Medicine at Rush University Medical Center in Chicago, working with my PI, Dr. Jitesh Pratap. The title of my talk today is RUNX2 promotes microtubule stability for survival of bone metastatic breast cancer cells. Can everyone see my slides? Bone metastasis is a frequent complication of breast cancer. There's an estimated 250,000 new cases of breast cancer diagnosed yearly, with bone metastasis being a major concern. Roughly 65 to 80% of patients develop metastasis during the course of their disease, resulting in pain, pathological fracture, and loss of quality of life. One of the biggest issues related to metastasis is that there's no currently targeted therapy available, resulting in reduced long-term survival. We see in the schematic shown below the metastatic process, whereby cells dislodge from the primary tumor and travel through the circulation to go to the preferred sites of metastasis in the bone, the brain, and the lung. The metastatic process is actually quite stressful on the cells, raising the question of how did these cells survive? We see in the radiograph shown here the typical osteolytic lesions highlighted with the arrows in the bones of metastasis patients. This raises the question, what mechanisms of survival are helping these cells cope with the stress of metastasis? One such mechanism of survival is autophagy. Autophagy is a process whereby cells enzymatically degrade defective organelles and proteins into subunits for synthesis, typically initiated in response to nutrient and hypoxic stress. Autophagy is marked by the rate of synthesis of LC3B2 protein and its subsequent degradation in a process known as flux. We see here in response to AMPK, hypoxia signaling among others, the formation of a double membrane vesicle engulfing cytoplasmic contents. This vesicle, known as an autophagosome, traffics towards the lysosome along a subset of stable acetylated microtubules for degradation. This vesicle is marked by LC3B2 protein, and high levels of autophagy have been linked to invasion, survival of metabolic stress, and metastasis. When we began our study, we asked the question, what is the status of autophagy during bone metastasis of breast cancer? Using an MDA231 model of breast cancer metastasis, cells were intracardiacally injected into mice, and tumor cells were isolated via GFP sorting from the bone. We see here in this H&E staining the interface between the tumor and the bone. One interesting point to note about autophagy is that counterintuitively, high levels of autophagy marker LC3B2 can be due to lack of turnover rather than induction, and low levels appear due to high levels of turnover. We see here in this schematic a stable microtubule, which is marked by an acetylation at lysine 40 of alpha-tubulin, which serves to recruit molecular motors kinesin and dynein to traffic the autophagosome towards the lysosome for degradation. Comparing autophagy markers LC3B2 and secondary marker P62 between parental and bone-derived cells, we see reductions in both LC3B2 and P62 in bone-derived populations. When examining the level of turnover using the lysosomal inhibitor chloroquine, we see parental cells show decreased accumulation relative to bone-derived cells, suggesting higher levels of turnover in bone-derived cells. The question that exists is what mechanisms are regulating this enhancement of autophagy? One mechanism that we came across is the transcription factor RUNX2. RUNX2 is a master transcription factor essential for bone development and is typically observed in the hypertrophic chondrocytes and in osteoblasts. One piece of information of note is that RUNX2 has been shown to be associated with the development and progression of bone metastasis and is aberrantly expressed in our MDA231 model. As we began to investigate the role of RUNX2, we initiated a study of the flux in bone-derived cells expressing either control or RUNX2 siRNA stably integrated. When examining LC3B2, we see accumulation of LC3B2 with RUNX2 silencing. And following treatment with lysosomal inhibitor chloroquine and secondary inhibitor baflamycin, we see robust enrichment, nearly 14 and 11 times in control cells with minimal accumulation and knockdowns, suggesting high levels of LC3B2 with RUNX2 silencing are due to lack of turnover rather than lack of induction. We also assess the status of acetylated alpha-tubulin, and we see reductions in acetylated alpha-tubulin with RUNX2 silencing. To corroborate these findings, we initiated electron microscopy studies, and we see here in control cells low levels of autophagosomes highlighted here in the yellow. However, with RUNX2 silencing, we see accumulation of autophagic vesicles marked by the red arrows with the zoomed in image shown here. We also used transfection techniques to do imaging, and we see that in control cells transfected with RFP-LC3 plasmid, we see low levels of signal. However, there's significant accumulation with RUNX2 silencing. While typically viewed as a transcription factor, we wanted to examine what possible mechanisms by which RUNX2 is promoting autophagy. we initiated a PCR array and tested 84 genes related to initiation, phagophore formation, lysosome activity, apoptosis, and other regulating factors to compare between control and RUNX2 knockdown bone-derived cells. We were able to identify one target as being different at the transcriptional level, transglutaminase 2. To verify this difference, we tested the protein level and we saw significant reduction with RUNX2 silencing. To determine the contributions of transglutaminase 2 to our model, we treated cells with the transglutaminase inhibitor LDN-27219 and assessed acetylated alpha-tubulin levels. We see here in the Western blot shown before that, similar to the previous Western blot, acetylated alpha-tubulin is reduced with silencing. However, following treatment, there is no significant change between the control and the knockdown in their untreated state, suggesting that transglutaminase 2 is not playing a role in our observations. This brings us to our hypothesis that RUNX2 is promoting cytoskeletal stability to enhance vesicle trafficking for autophagy and secretion in bone metastatic breast cancer cells. We see here in the schematic the autophagosome marked by LC3B2 protein trafficking along this acetylated microtubule to promote autophagy, turnover, survival, and motility. Furthermore, near and close proximity to the microtubule, we see positive regulator of alpha-tubulin acetylation alpha-TAT1 and negative regulator HDAC6, with RUNX2 serving an unknown role. It should also be noted that RUNX2 has been known to be associated with microtubules. The significance here is that RUNX2 is currently viewed as a transcription factor, showing nearly 70% nuclear localization, with a small cytoplasmic fraction of 30 to 40% serving an unknown role. Understanding the cytoplasmic function could help link transcriptional activity with functional roles, and understanding the relationship between RUNX2, autophagy, and alpha-tubulin acetylation could potentially offer new therapeutic targets and explain the failure of HDAC inhibitors in the clinic. Interestingly, high levels of RUNX2, acetylated alpha-tubulin, and autophagy have been independently linked to the development, onset, and progression of aggressive migratory and metastatic cancer. A little bit of background about acetylated microtubules. Acetylated microtubules are long-lived and have a half-life on the order of minutes compared to seconds, which is observed in typical microtubules. Acetylation has been shown to promote kinesin recruitment and enhance trafficking speed, as well as to allow compliance and resistance to mechanical stress. It is positively regulated by alpha-TAT1, with HDAC6 and sirtuin-2 serving as negative regulators. We see here that alpha-TAT1 enters the lumen of the tubule to produce acetylation, and these microtubules are able to bend and flex in response to force without buckling and breaking, as in the case of non-acetylated tubules. The question that was this produced was, how does RUNX2 enhance acetylation? We initially surveyed the protein levels of positive and negative regulators of acetylation, and we saw that sirtuin-2, HDAC6, and alpha-TAT1 were not changed with RUNX2 silencing. While alpha-TAT1 inhibitor wasn't available, we examined inhibition of sirtuin-2 and HDAC6. To corroborate our previous findings, again, we see nearly 50% reduction in acetylation with RUNX2 silencing. Using the sirtuin-2 inhibitor AK7, we see no significant change in acetylation with treatment. However, using the pan-HDAC inhibitor TSA, we see a robust induction of acetylated alpha-tubulin, suggesting that sirtuin-2 doesn't play a significant role in regulating alpha-tubulin's acetylation, and the primary player is HDAC6. It should be noted that we were able to obtain similar findings using the HDAC6 specific inhibitor tubacin, as well as using HDAC6 siRNA to knock down HDAC6. In the schematic shown below, we see the preferences of HDAC6 and alpha-TAT for fractions of the microtubule cytoskeleton. Alpha-TAT enters through the lumen of a polymerized microtubule to induce luminal acetylation of alpha-tubulin. Conversely, HDAC6 shows preference for oligomers of alpha and beta tubulin and deacetylates following depolymerization, suggesting that low levels of acetylation correlate with low levels of polymer mass. As we began to continue this, we wanted to examine the architecture of the acetylated microtubules within our model. We stained for control and RUNX2 knockdown cells for acetylated alpha-tubulin, and we see here in these images shown in black and white to highlight morphology, a strong perinuclear ring of acetylated alpha-tubulin with minimal disruption and lack of banding. We also see in radiating microtubules even staining. However, with RUNX2 silencing, we see significantly lower staining intensity, disruptions in the perinuclear ring, and lower levels of radiating microtubules. Hypothesizing that low levels of acetylation correlate with low levels of stability, we treated cells in the presence and absence of the vinca alkaloid vinblastin, which is able to depolymerize the acetylated fraction of the tubulin cytoskeleton. We see in control cells some disruption and decrease in signal following treatment. However, structure remains, whereas with RUNX2 silencing, we see already low levels of signal, and following treatment, we see the formation of spiral aggregates, which are known to result from depolymerization of microtubules. We also assessed the status of the polymer mass and were able to isolate microtubule polymer mass from untreated cells. Plotting the microtubule polymer mass, we see robust levels of both alpha tubulin and acetylated tubulin in control cells, with no acetylated alpha tubulin and knockdowns and low levels of alpha tubulin. These results suggest that the primary form of acetylated alpha tubulin with RUNX2 knockdown is in the oligomer form, awaiting deacetylation by HDAC6. Quantification of our microscopy shows significant reduction in intensity with RUNX2 silencing, as well as in control and in vinblastin-treated conditions, highlighting the lack of stability. The question that exists now is whether this instability is a consequence of lack of transcriptional support. Using viral vectors, we transduced bone-derived RUNX2 MDA231 knockdown cells with empty vector, wild-type RUNX2, and transcriptionally inactive DNA-binding mutant RUNX2 constructs. Similar to previous findings, we see lack of a perinuclear ring and disruptions in the acetylated microtubule cytoskeleton in the empty vector group. However, in response to both wild-type RUNX2 vector and DNA-binding domain mutant RUNX2 vector, we see the emergence of the perinuclear ring and evenly acetylated microtubules with quantifications yielding significantly greater staining intensity in both wild-type and DNA-binding mutant expressing cells. Furthermore, following vinblastine treatment, we see that wild-type expressing cells and DNA-binding mutant expressing cells are able to maintain some level of microtubule polymer mass relative to empty vector control, with the quantification depicted here. These results suggest that reintroduction of wild-type and DNA-binding domain mutant RUNX2 to silent cells can rescue the acetylation. DNA-binding domain mutant and wild-type expressing cells show decreased sensitivity to vinblastine relative to empty vector. Furthermore, transducing these vectors into cells expressing control siRNA also raised acetylation to levels greater than that in the empty vector control, suggesting that transcriptional activity is not required to promote acetylation and stability, and that these functions are independent of transcriptional function. To examine the status of regulators of the microtubule cytoskeleton, we conducted a second PCR array examining 84 genes related to microtubule-associated proteins, actin-related proteins, cytoskeletal signaling and dynamics, among others. Examination of these 84 genes yielded one target, an actin-related protein moisin, which showed slight upregulation at the transcript level. When we went to verify this at the protein level, we saw that while it was elevated in RUNX2 silent cells, the difference was not significant. We wanted to see how our findings correlate with the clinical case of metastasis. Using a small library of patient-derived primary breast cancer biopsies and unmatched bone metastasis biopsies, we stained for acetylated alpha-tubulin. We see here the staining in the cancerous tissue of the primary tumor and enhanced staining in the metastatic tumor, with the bone tumor interface shown here. These images are shown here as controls for negative control and what constitutes a 3-point positive on our staining scale. And we see that unmatched metastasis shows significantly higher 3-point positive staining cells for acetylated alpha-tubulin. These results suggest that acetylated alpha-tubulin and RUNX2 may serve as biomarkers for bone metastasis. Furthermore, it's notable that microtubule targeting drugs are commonly used as chemotherapeutic agents in the clinic. However, not all chemotherapeutic agents are able to target the acetylated fraction of microtubules. Conversely, the commonly used agent docetaxel inhibits growth of tumor cells by stabilizing microtubules and actually results in hyperacetylation of the microtubule cytoskeleton. RUNX2 and acetyl alpha-tubulin levels may help clinicians better predict which therapeutic agent a patient's tumor will respond to. These observations in breast cancer got us interested in what is the status of microtubule acetylation in normal cell types that express RUNX2. We began using microscopy to stain primary calvarial wild type and RUNX2 null osteoblasts for acetylated alpha-tubulin with and without vinblastine treatment. In wild-type cells, we see robust staining with a strong perinuclear ring and evenly stained microtubules, which, while disrupted following vinblastine treatment, maintains a fair amount of stability. However, RUNX2 null osteoblasts show disruptions in the perinuclear ring, tangles in acetylated microtubules, and almost complete disruption following vinblastine treatment. At baseline, RUNX2 null osteoblasts show diminished staining intensity, albeit not to a level of significance. However, following vinblastine treatment, there's a significant difference in intensity between wild type and RUNX2 null osteoblasts. These results in culture intrigued us and led us to do some immunostaining on a cohort of hind limbs from wild type and RUNX2 conditional 1 collagen 1 Cre knockout. We see here in the figure below staining for acetylated alpha-tubulin with significantly higher number of positive cells in wild type limbs relative to conditional knockouts. We see again in the schematic shown below, RUNX2 promoting the acetylation of tubulin instability to allow for recruitment of kinesin and dynein and trafficking of the autophagosome. This schematic is notable as silencing of RUNX2 as well as silencing of the autophagy pathway have been shown to cause defects and lack of skeletal development in animal models. The second site of RUNX2 expression that's observed in the skeleton is in the hypertrophic chondrocyte. We derived wild type and RUNX2 null primary chondrocytes from mice and stained them for acetylated alpha-tubulin. We see in wild type cells robust staining with diminished staining intensity and disruptions with RUNX2 silencing. In these images in black and white to highlight morphology, we see parallel linear bundles in the perinuclear region of acetylated alpha-tubulin, which are evenly stained, whereas RUNX2 null chondrocytes show disruptions in this perinuclear acetylated alpha-tubulin, shown here and here, as well as show banding with localized loss of signal. Quantification yielded a significant decrease in staining intensity with RUNX2 silencing. That led us to our next experiment, whereby we hypothesized that these defects can be treated by chemically stabilizing the microtubule cytoskeleton. We treated wild type and RUNX2 null chondrocytes in a dose-dependent manner with the chemical stabilizing agent docetaxel. While both cell types show increase in signal intensity with treatment, wild type cells show a dose-dependent increase in bundling, with 10 micromolar dose resulting in parallel linear bundles of acetylated microtubules in the perinuclear region, whereas RUNX2 null chondrocytes show high levels of signal. However, the lack of organization remains, suggesting other factors are required to produce these bundles. In conclusion, we see the autophagosome marked by LC3B2 trafficking along an acetylated microtubule that's compliant and resistant to stress to allow for autophagic flux, resulting in subunits for synthesis to promote cell survival and motility. We also see alpha-TAT1, HDAC6, and RUNX2 in close proximity, working to promote the stability and acetylation of these tubules. In conclusion, we see that RUNX2 silencing does not alter expression of critical autophagy or cytoskeletal regulators. We see that RUNX2 promotes stability independent of transcriptional activity. and that key regulators of acetylation are unchanged with silencing. We see that silencing enhances sensitivity to vinblastine, and that patient samples show significant increases in acetylated alpha-tubulin and bone metastasis versus primary tumors. In the future, as we continue our project, we're working to isolate large quantities of microtubule polymer mass to be able to send for proteomic analysis via mass spec to determine which proteins are differentially isolated in the polymer mass of control and RUNX2 knockdown cells. We are also working on developing an in vivo model of microtubule targeting drugs in bone metastasis to selectively target the acetylated fraction and determine their contribution to the development and progression of disease. As I conclude, I'd like to thank the ASBMR and the organizers of the Spotlight Series. I'd like to thank the members of our lab, my mentor, Dr. Jitesh Pratap, Manish Tandon, our technician, Marcus Winogrodsky, and other students in our lab, Shreya Patel and Waddell Holmes. I'd also like to thank other members of our university community, our department chairperson, Dr. Sumner, for help with obtaining bone samples in microscopy and histology, Dr. Subin Chubinskaya, for providing us with cartilage samples and chondrocytes from their mice, the Prodescu Lab, and the Department of Pathology. I'd also like to thank our collaborators, Drs. Stein and Leon at the University of Vermont, for providing mice and viral RUNX2 constructs, and Dr. Javed at the University of Alabama for sending us blocks of mouse tissue for histology. That's the end of my presentation, and I'm going to open the floor to any questions you might have.
Thank you, Ahmed. That was a very nice presentation. Can you hear me okay?
Yeah, I can hear you. You're welcome.
Okay. I had some questions about your animal knockout model. So how old were the mice? And then also when you talked about the bundling or the lack of bundling following your treatment, what significance does that have?
The animal model, off the top of my head, I can't recall the age of those mice that we used for the histology. As for the bundling, we don't have a quantitative measure of the bundling yet. However, in looking at the acetylated microtubules across the MDA cells, osteoblasts, and chondrocytes, one of the most frequent things that we observe is that there's a strong perinuclear bundle of acetylated tubulin in the wild type cells that's diminished in the RUNX2 null cells. It seems that the acetylated microtubules are not widespread throughout the cell, and that if you do overlaying of the whole cell versus where the acetylated fraction is, the acetylated fraction, it remains condensed within the perinuclear region.
So for your animals, were they developmental age? I know you don't know the exact age, but were they in stages of development?
I think they were like getting close to where the growth plate was sealing, but not completely sealed.
And did you notice any skeletal defects or any defects in the chondrocyte or the tissue itself?
No, the RUNX2 conditional knockout mice, their osteoblasts don't develop properly, but in our staining, we were staining for just the level of acetylation rather than markers of development or anything like that. I can share one piece of information here. We actually differentiated osteoblasts in culture from primary calvarial osteoblasts. And what we see is if you differentiate these cells in culture, as the cells differentiate, there's a robust induction of autophagy and alpha-tubulin, acetylated alpha-tubulin, that coincides with the induction of RUNX2. As you go out towards osteoblast to osteocyte transition, this induction begins to level off, reaching its peak at somewhere between day 14 and day 21. However, it doesn't go down, suggesting a physiological role for the acetylation and autophagy during skeletal development.
Can I ask you a question, Ahmad?
Yes.
It's a very nice presentation. Do you know which amino acid in alpha-tubulin is getting acetylated? Lysine?
Lysine 40.
And then which the lysine acetyl transferase, the enzyme act on that?
It's alpha-TAT1, acetyl alpha-tubulin transferase.
No, acetyl transferase.
Yes,
So that's the same, that's behaved as an acetyl transferase?
correct. It's known as alpha-TAT1, and it also goes by the name MEC, MEC 17.
Mhm. Okay, thank you.
You're welcome.
I have a question. So is there any sort of information that suggests that these triple negative breast cancer cells don't, like what you guys are talking about looking at bone nets, are there any differences with RUNX2 and the tubulin in nets that go to lung or brain? Or have you been able to really take a parental MDA and maybe compare it to your bone tropic and see if any of the hits from the pathway you're interested in come up with those cells?
We have the cells from the other metastatic sites, but when we began the project, the comparison was against parental and bone. And when we saw that this was enriched in the bone, we did the experiments in the bone-derived. But I agree, comparing against the other metastatic sites is something that's very worthwhile. But I'll share one piece of information that I got from one of my references. When they looked at autophagy across breast cancer types, the triple negative patients in the cohort had the highest levels. Also, when they looked at them stratified by the metastatic sites, the bone metastatic patients had the highest levels. Keep in mind there's regional differences, between the bone, the lung, and the brain. And those differences might alter the metabolic programs of these organ-specific clones. One other piece of information is that in normal cells, autophagy needs to be induced. However, due to MAPK mutations in KRAS and BRAF, autophagy becomes deregulated. In the case of the MDA, which expresses both of those mutations, the autophagy is functioning at an extremely high level, irrespective of the nutritional status, and it's further enhanced following the metastasis.
Hi, Ahmad. This is Paula de Bettifaz. Very nice presentation. I have a couple of questions for you. And maybe it's just because the picture that you show, but it looks to me that when you show the RUNX2 knockout, either osteoblasts or chondrocytes, that the effect of the alpha-tubulin was way more evident in chondrocytes than in osteoblasts. Is that was just the picture that you show, or you also saw quite a dramatic effect in the osteoblasts? And then my second question was I think you showed that when you treat your, and I forgot it was chondrocytes or osteoblasts, the RUNX2 knockout with, I believe was docetaxel or something like that. You could actually the alpha tubulin was, the acetylation of tubulin was reestablished, or the bundle were established, something like that, right? I forgot exactly the details. So my question is, does that also induce a change in the functional effect? Because, you know, we know that when you knock out ranks 2, you don't make osteoblasts, right? You have only a cartilaginous skeleton. So you know, can you almost like treat or rescue your ranks 2 knockout just by giving whatever treatment you were giving? Have you tried that?
Actually, these experiments that you're asking about are something that we have currently in progress.
Oh, I see.
I've done this with the MDA, with the breast cancer cells, and I see some interesting findings. Knockdowns is unable to target the acetylated fraction. So when you treat with knockdowns, as soon as you remove the drug, you see actually enhancement of acetylated tubulin following removal of the drug. And that enhancement coincides with an increase in turnover of the autophagosomes. During vinblastine treatment, as soon as you remove the vinblastine, the acetylation is completely lost. And if you let this continue over time, it's actually unable to recover. In terms of Taxol, we tried to measure the flux through the autophagy system with Taxol treatment. And what we saw is that while it stabilizes microtubules, it causes non-specific stabilization, such that everything becomes frozen and the turnover is not restored. While the trafficking is dependent on the acetylated fraction, some of the earlier stages of autophagy are dependent on the dynamic fraction, and the balance between the two fractions is critical for maintaining a functional pathway.
Nice, thank you.
You're welcome. I'd like to make one more comment though. Chemical treatment may help enhance osteoblast differentiation. However, what chemical treatment can't compensate for is the lack of transcriptional support that occurs with runx 2 null osteoblasts or chondrocytes.
I just had another thought in my head. Have you tried any sort of functional experiments with some of these, with the parental cell NDAs, invasion or migration? Because if the whole idea is that there's some sort of link to bone metastasis with some of these treatments or some of these disruptions that you're doing, or inducing autophagy in a parental cell and seeing if they're more invasive or migratory, that could be a nice just in vitro experiment to support what you're showing. And then also with all of your different knockouts and drugs, that would be kind of interesting to see if that fits.
I did that with autophagy, and I did it in control and runx 2 knockdown parental cells, as well as cells expressing the control in the runx 2 SH after they were pulled from the bone. And what I see is that the runx 2 silencing significantly reduces the migration compared to their corresponding control. But if you treat with chloroquine to inhibit autophagy, you actually see a significant reduction in the migration of the cells expressing the control. One of the things that autophagy is thought to be playing a role in is the turnover of focal adhesion complexes, which is important for movement of cells. And that to inhibit autophagy, they're unable to cleave and reattach. There's another paper by Amanda Boggs that the acetylated tubulin is important for the formation of microtentacles in breast cancer cells to help them attach to their substrate and be able to move and work their way through the environment. And the introduction of a plasmid that has that acetylation site mutated helps lessen their ability to attach and migrate. I hope that answered your question.
Yep.
Any other questions today?
Great, thank you so much, Ahmed, and thank you everyone for joining and asking such great engaging questions. I would just like to remind everyone that the series is occurring bi-weekly, as I mentioned, and the next session will be November 19th, which is a Thursday at 3 p.m. Eastern time. And the presenter list will be published shortly for the remainder of the dates with the topics as well. So we hope to see you next week, in 2 weeks, and for the rest of the sessions as well.
Hey, Lauren, sorry, I have a quick question.
Mhm.
Is there different Zoom link, or because in my calendar I already have the bi-weekly Zoom link? Do I still need to register every time, or I can just go and
It
use?
should be the same recurring link.
Okay.
So
Because
it
it is already pre-populated my
Mhm.
calendar, so I just didn't know if I had to re-register or I just
Nope,
use the
it
same.
should, yeah, the recurring calendar invitation should work for all the meetings moving forward. And then when we finalize dates in 2021, that will be a different link, and we'll circulate that once we have those confirmed.
Okay, wonderful.
All right, thank you all for joining. Enjoy the rest of your afternoons.
Thank you so much for this opportunity. You have a nice
Thank
day.
you again.
Yep,
We
I
enjoyed
appreciate
conversation.
your questions and comments. They were very helpful.
Good. All right, bye.